Hi, I'm Hermine Maas, and this is the second video in the Heterogeneity series on gene-by-environment interaction. We'll talk a bit more about genotype by environment interaction models and theory, and a lot of this is described very well in the 2002 paper in Twin Research by Purcell. The application of these G/E models was facilitated by the implementation of definition variables by Mike Neal in ClassicMX sometime in the 1990s, so predating this paper. Sean does a great job of talking about gene-by-environment interaction models and the various possible extensions that are possible of this basic model. And we'll talk about those in a minute. But first, let's point out some of the terminology here. The standard G/E models in Sean's paper will be referred to as A by M, where A refers to the additive genetic variance and M to the moderator, as we can, of course, moderate either the additive genetic variance components or the shared or unshared environmental variance components, which we would call C by M or E by M. And each of these different types of effects fall under the general class of genotype by environment or G/E effects. Sean also spells out the definitions, again, very clearly, where genotype by environment interaction, or G times E, is conceptualized either as the genetic control of sensitivity to different environments or environmental control of differential gene effects, where RGE, or the correlation between genes and environment, represents the genetic control of exposure to different environments or the environmental control of gene frequency. He also points out that if these effects exist but are not included in the model, we end up with biased parameter estimates of the A, C, and E variance. And it works as follows. If we ignore interaction of the A by C type, that would act like additive genetic variance. In other words, the A component would be overestimated when A by C interaction is part of the real world of the phenotype. A by E interaction acts like E, the correlation between A and C acts like C, and the correlation between A and E acts like A. So as I've indicated before, one or more environmental moderators may be either binary or continuous, and we'll go more into the continuous case in this presentation. But they can also be the same for members of a twin pair or differ between twins in a pair. And that has different implications on the kinds of models that we can fit. Now, the most basic G/E interaction model involves a moderating variable that implies that genetic effects either increase or decrease as a linear function of the moderator. And I think this becomes clearer when we look at it in the context of the biometrical model. And you see here that a linear A by M interaction implies that the additive genetic value is a linear function of the moderator. Now, this results in a variance that is a quadratic function of the moderator under linear interaction, because we typically square the effects of the additive genetic value. Again, as noted before, one or more environmental moderators can interact not only with the genetic effects, but also with the residual environmental effects, either the C or the E effects. And so as you see in this figure 2 here, which is the classic Purcell representation of the G/E model, we have for each of the paths, the unmoderated component A, C, or E, and then the moderated components, Bx times the moderator, By times the moderator, or BZ times the moderator. One or more environmental moderators may have not just linear effects, but also nonlinear properties, as indicated in these figures here, where the moderator may have a greater effect on the extremes than on the mean of the distribution, or vice versa. One or more environmental moderators may show scalar or qualitative interactions. We will talk primarily about scalar interactions, which, as explained by Sarah Medland in the sex limitation video, we refer to testing whether the quantity of the variance components or the magnitude of the variance components is different in different groups, or in this case, for different values of the moderator. But similarly, we could also test whether there are qualitative effects, or different genes that interact with the moderator. But this can only be done when the moderator differs between the members of a twin pair. Finally, one or more environmental moderators may also be correlated with the genetic effects acting upon the trait, which then allow us to test for gene-environment interaction in the presence of gene-environment correlation. And this, as you can see in this diagram, is done in a bivariate context where we include the moderator as a dependent variable. And so we will talk more about that when we move into the multivariate space. So a little bit more about how we go about testing the effects of a continuous moderator. And we do that typically using a definition variable approach. And so here, the question is no longer, is the magnitude of the genetic and the environmental influences on the trait the same in two groups, but now across all values of a moderator, for example, age. In other words, is that beta coefficient that we multiply with the variance components significantly different from 0 or not? And as I said, age is a very typical example of a continuous moderator, and one where we, of course, also can be very sure that it's environmental and doesn't have a genetic influence as well, which cannot necessarily be said about a variety of other variables that have been considered as moderators. So when we talk about additive genetic effects moderated by age or age by A interaction, of course, we can extend those types of models with also testing whether age moderates the C and the E variance components. Now, other classic examples are where either the A effects or the A, C, and E effects are moderated by socioeconomic status. And here, the classic examples are moderation of the additive and shared and unique environmental variance effects on IQ or intelligence by Eric Turkheimer and the Tucker-Drobe and Bates paper. So this is really a classic example of G by E with a continuous moderator, where Eric shows that the heritability of IQ seems to change as a function of the value of socioeconomic status, and so increases with higher SES. And correspondingly, the proportion of variance explained by C and E appears to decrease with SES. This result was replicated about 10 years later by Tucker-Drobe and Timothy Bates in a mega-analysis, where again, they find that the genetic variance is increasing whilst the shared and the non-shared environmental variance decreases as a function of socioeconomic status. They also show a variety of ways in which we can plot this appropriately. So as indicated, definition variables that were implemented first in classic MX and now also in OpenMX were critical here to fit these types of models. And what are these definition variables? These are variables that may vary per subject or per pair and that are not dependent variables. They are typically represented in a path diagram as a diamond, and we can use them not only to model the main effects of covariates on the means, but also to model changes in the variance components. So when we first model them on the main effects, then it's really like a regression of the phenotype on the covariate, where the residual variance can then be further partitioned into sources due to A, C, and E. Now, in a second step, we might be able to also test whether the ACE variance components also change as a function of the variance components. And we'll show how that goes in a series of path diagrams. First, the basic representation of definition variables in a path diagram. The top diagram just shows the path diagram for the main effect of our variable of interest x, the dependent variable, where the main effect is indicated by the triangle. We multiply 1 by the path coefficient μx to get the main effect. We can add a moderator to the model as a measured environmental variable that also has an effect on our dependent variable estimated by beta x, and that we can add to the main effect in figure 2. The third figure here is just an alternative way of representing that same model, but now using a definition variable, which makes it explicit that we use this moderator not as a dependent variable, but just as a way to multiply with the regression coefficient to estimate the moderating effect of the moderator on our dependent variable x. So back to our standard ACE model for twin data. This is the basic version that's where we don't have any moderating effects. We do have estimates for the mean, mu, and for the 3 variance components, in this case, A, C, and E. And this is a path coefficients model. As I've noted earlier, when we include moderation, the assumption is that the moderation is a linear function of the additive genetic value, and therefore we moderate the path and not the variance component. So before we go there, we are first going to add the moderator as a main effect on the mean by adding our definition variable using a dummy circle in there, so that we can multiply the beta coefficient with the actual value of the moderator, in this case h, to take out the effect of h on the mean of the phenotype before partitioning the remainder of the variance into A, C, and E components. Of course, the focus here is on G by E. So in this case, we've now added an arrow from the additive genetic component through the moderator to the phenotype, so that we can basically partition the additive genetic variance in that portion that is not moderated, the A portion, and then a portion that is moderated by, in this case, H, that is the AI portion that gets multiplied by the value of H for your particular twin pair to estimate whether or not the additive genetic variance changes linearly as a function of H. As noted before, not only can we potentially moderate the additive genetic component, but also the shared and the unshared environmental component. And so here we've added 2 additional paths, CI and EI, indicating potential moderation of the C and the E component. And as you see in the expectation now, not only will we add the squared components of A and AI, C and CI, E and EI to the expectation of the variance and the covariance, except for the E component, of course. But we square the A plus AI times H component so that also the covariance between A and AI gets included and contributes to the variance of the phenotype. Now, for those of you familiar with Purcell's paper, you may think that this path diagram looks quite different from his. Here they're side by side. They're really the same model, but instead of having both the unmoderated and the moderated component on one part, we have separated that out in the diagram on the left, as we can independently estimate the contributions of the moderated and the unmoderated variance components on the phenotype. And with that, that's the end of this presentation. And I just wanted to thank those of the faculty that have contributed most to the theory of this type of application: Linda Neves, Mike Neal, Pak Sham, and Sean Purcell. Thank you very much.